The dissection of the calcium signals at a subcellular resolution is one of the most important steps for decoding intracellular calcium signals that determine the output biological phenomenon. This protocol describes a new calcium imaging method that enables the monitoring of the very moment of the calcium influx and calcium release.
This protocol is applicable to all cell types that allows the expression of genetically encoded calcium indicators. We believe that our method has the potential to be expanded to the dissection of calcium signals at subcellular resolution in living animals in the future. Helping to demonstrate the procedure will be Matsumi Hirose, a technician from our laboratory.
To begin this procedure, place an 18mm diameter glass cover slip in each well of a 12-well plate. Use sterilized water to prepare 12.5 milliliters of a 0.04% PEI solution for each 12-well plate being used. Add 1 milliliter of the PEI solution to each well, and ensure that there are no bubbles underneath the cover slips. Incubate overnight in a carbon dioxide incubator at 37 degrees Celsius. The next day, use an aspirator to remove the PEI solution. Add 1 milliliter of sterilized water to each well, and shake the plate so that the PEI solution between the cover slip and the plate is washed.